Hello, welcome back to the Computational Genomics Lab. We are now getting ready to analyze our RNA-seq pipeline output. So you've run the RNA-seq pipeline, and you might be wondering, where's my data? Well, you had that output directory in your run.sh from your RNA-seq script or your nextflow script, and that's where you want to go into that directory, and you have a bunch of results there. And we're going to first work on the counts. And what counts are the amount of reads there were per gene in the genome. So what the nextflow pipeline did is took all those raw data, reads, aligned them to the genome, and then counted them up. And there's a bunch more detail in the Github file for this here. And let's just go, because this is how we're going to be proceeding through the rest of the class, is working from GitHub into RStudio. And this is the Rinn class GitHub for the class, and we're in 05 here in lessons. So if we go back up here, this is what you'll see in your GitHub. You should have cloned this by now and have it just like this. 05 RNA-seq in RStudio, and we are doing 03 skills in reading in our data. So we're going to learn our skills as we start our analysis, and we're going to go really slow. We're going to go over these things a lot. They'll become more routine, but for now we're going to learn some R functions, some more RStudio functionalities, and all while trying to analyze our RNA-seq data to find novel genes that are changing due to doxycycline exposure. So I'm going to go to my RStudio. Yours might be local, and we sort of had a video going over that and how to install packages. Today's lesson won't need any packages because we're going to do it all in base R. And just to remind you here, this is where you are. So if we go to class and then lessons, we're in 05, and we just pulled up 03, R skills reading in data here. Okay, so that's all you would do. And now this code will run from here and produce things in our environment, objects, and we're going to go over all that. But the thing that's really important is that you set your working directory. And that's here with this cog button. It has this thing as set as working directory. This is really important as a first step anytime you open up RStudio, even if you know you're in the right place, because whenever you save a file, if you were working somewhere else or your working directory is somewhere else, it's going to save it there, and you might think it's lost, etc. So it's just a good habit to do that right away. And then again, the GitHub, the .rmd files on GitHub that you're loading in here all have way more detail than what I'm going to go over in the video. And the goal of the video is just to see it run through, and you can go back and get more detail from here. And so the program that did all of the aligning, took all the reads and aligned them to the genome, was called STAR. And the program that counted those reads over the genes is called SALMON. And that's where your output is, and you can see that here that I called mine pipeline output. You might have called it the same thing, but it was in that run.sh. So if you're not sure where it is, you can go back to that run.sh and find it there in the star salmon folder. And what we really want here is salmon.merge.genecounts TSV, which means it's tab separated. This is just a table, like an Excel file, of all the counts for each gene. And that's what we're going to put into DESEQ to do our statistical analysis. So we're getting really close to doing the fun part, but we're going to learn some R skills to prepare the data to be able to do that. Okay, first things first, let's read it in with this function read.table. If you want to know more about read.table or any function we go over, you can put a question mark in front of it and run that, and you'll get a help menu down here that'll tell you what things you can do and all kinds of other things. And that's in the help panel in this down here, and we're going to go back to our files of where we are. Okay, so if you want to know more, but it's really simple. It's just going to read in a table, and it has some things you can tell it. So this needs to first know where is the table, and it's in quotes because it's a file path. It's just not an actual thing it's going to do. It's just going to say, tell me where to go, and that's in quotes. That also gets a little confusing, and it'll get easier as we go. There's a header. That's true because our counts matrix has row name, or each column has a name, and each row has a name. So we're going to say the header is true and that there are row names in the first column. Okay, let's read in the table. And boom, it shows up in here in the environment. A couple things to notice right away is we have 55,401 observations, that's how many rows we have, and 16 variables, that's how many columns we have. This is a quick way to check how things are changing as you do more and more code. Okay, so we can either click on this and view it here, and what we see here, and we're going to get back to this, is this long string of things. That's the gene ID, this ENS number here. But notice it's in the black of the table. It's in the metadata, if you will, and that it's keeping track of what that row's name is. But in this case, gene name here is actually in the data frame itself. And then the columns are 0 time point replicate 1, 0 replicate 2, etc., etc. Okay, cool. So now another way to do that is run an R function called view. And anytime you run an R function, you're going to put in parentheses what that function is going to work on, and the question mark will tell you what things it knows to work on. And that's really what functions do, is they say, give me your input, I'll do my thing to it, and give you the output. So we can also run view, which is just simply going to open that, same thing. We see the gene ID is this long number, and gene name is more of like a letter thing you can understand. And we'll be using those quite a bit. Okay, so again, more detail in here. We've loaded our file, and we need to, sorry, I have the wrong file open. We need to make an archive of what genes we analyzed. So different pipelines, different annotations. You put a GTF file as an annotation, you're going to have different gene numbers and levels and stuff. So we want to archive that with this object we're going to create here called G2S, which stands for gene ID to gene name, or gene ID to gene symbol. But it's just going to be 2 columns that has that long string and the gene name, and that'll be a really handy archive of all the genes we looked at just to start with. So first thing, we make a new object name in front of this arrow thing here. This means anything you do on the right side of this arrow is going to be put into this object's name here. And if you name the same thing and run multiple functions on it, it's going to keep the same name, but it's going to keep changing. And so sometimes it's good to change this object's name every time you do something to it, and we'll do that in just a second. But for now, we're going to run this data frame function, and that's just simply saying I'm going to make a table like Excel would in blank Excel sheet. And you're going to say the first column is the parameter this function needs is called gene ID, and that is equal to row names. And what row names is going to do is go into that meta space that we saw here and grab those values for the counts matrix, and it's going to put them into a column called gene ID. And then similarly, we have this gene symbol column here, and it's going to go into the counts matrix. And then now you're going to see something funny. These square brackets here means I'm going to go into that file, and it's always rows and columns. And so there's nothing in this first value here. And so it's going to keep all the rows, which we want because those are the gene names. But we only want the first column, which is the gene name. Okay, so let's run that here. And so what we can see already is we have an object here called G2S, and it's just simply the gene ID's name and the gene symbol for any gene that had values in our data analysis. Cool. So now we can save this. Again, why that set working directory is so important. I made a results folder here. You can make a new folder with this button, create new folder, name it what you want, but I would go with results here. And we can use write.csv, an R function. Again, in the parentheses is what it's going to do. It's going to take this object G2S that's in our environment, and it's going to print it out as a CSV file or like an Excel sheet in this results folder. So let's do that, write.csv, go into our results, and we can see this G2S is in there now. Cool, we've made our first data frame. And again here, same thing, G2S is another way to make the same thing. And this is a little cleaner because you're saying run this function. Here's the first column, same thing. Here's the second column. And I want the values of this column to come from column 1 of counts matrix. Goes in with the square brackets and then says give me all the rows because there's nothing to sub-select there, and just subselect with the first column name. Okay, but now the problem is when we go do our analysis, we want to have only numbers in this table, and they need to be in an integer form. Right now they're numbers with decimal places and things like that. DESEQ is going to need us to round those, and we're going to do that in a second. But this gene name would cause us a problem because it's in character, but we've just made an archive that if we keep this in metadata and get rid of this column, it'll be all numbers in the table, and we can make it a matrix, and then we can put that into DESEQ. So let's do that. Okay, so we're going to make our counts matrix object. We're going to rename it again, kind of dangerous, but in this case it's okay. Counts matrix, we're going to square bracket, which means we're going to go into that object. We're going to take all the rows, and you see the -1 now means get rid of that first column, or take that column out. Okay, so now if we look at counts matrix, we don't have that gene name column anymore. Cool, and that's a really handy function you can do for anything, is just remove columns and index with the square brackets. Here's a couple other ways of doing it that are also marked down on how they work. But really it's all going to do the same thing. It's just to highlight that there's many ways to get to the same point. Okay, so next we are going to turn our counts table, now that it's all doesn't have any character values of the gene names, the column names are our time points and replicate. We're going to go into that and make it a matrix. Nothing's going to change. It's going to look the same. And we do, right now, we can see it looks like this. And once we run this, we can see that it's now added this bit here to let it know it's a matrix. And that's what we're going to need. DESEQ needs it to be a matrix to be inputted into it. Okay, one of the last things we're going to do is just round the values. So currently our counts matrix has decimal points, and DESEQ is not going to like that. So we're just going to round it. Using a function called round. Again, round is the function name. What it's going to round is inside the parentheses, and what we're going to rename it now is counts matrix rounded, because we did something to change the original file. We're going to rename it, and we're going to round the counts matrix. Let's do that, and then we can use this view function to view what just happened. Okay, cool. We can see we got rid of the decimal places. And you'll notice here that there's a lot of columns that have 0 values across the whole thing. And that's going to make data analysis not only bulky, but we really don't want to say we observed those genes if we didn't have any observations for them. So what our last step is, we're going to go find all the rows that equal 0. So if you sum that row and it equals 0, it means there was all the values at each time point were 0, and we're going to get rid of those. Okay, so we're going to call that now counts filtered. Again, there's a ton of step-by-step, much slower here, videos just to get you sort of familiar with it, and then you can go back and read in more detail. Okay, so we are going to make counts filtered, and we're going to take our counts matrix rounded, which is the object we just made here, and we're going to go into it with the square bracket index again. And we're going to use this function row sums. And now row sums is simply going to sum the values of the rows. It's really handy. We're going to use it later. But in this case, we're going to look for all the row sums that equal 0 or are less than 1, which means they had to be 0 because we don't have decimal places anymore. Okay, so we're going to run, now we're indexed into the counts matrix filtered, rounded, and we're going to run a function inside of that table, which is really cool. It's kind of like a macro in Excel, if you will. And so we're going to go into that table, and again, rows is the first thing, and then after the comma is the columns here. We're going to keep all the columns, whereas before we kept all the rows. This time we leave this blank, and that way we will keep all of the columns. And so we're going to go in and change our rows to make anything that we're going to sum them up, and we say if you're less than 0, or you have to be greater than 1 to be kept. Okay, so let's do that. And we now have count matrix filtered, and this is how we know it worked right here. This 45,000 elements is now what it is, and it used to be 83,000 elements. So essentially that means that half of our genes had no values and would have could influence our data analysis. Congratulations, you've done it. You've made your counts file ready for DESEQ. And now we're going to save it and see how handy this is. When I first learned this, I thought this was so cool that there's just a save function for R. So you just type save, and in the parentheses, what you're going to save is counts filtered. So it knows that's an object in your environment, counts matrix, our G2S, and then we have to tell it where to print that. And we're going to go to the file or in our results folder. I'm going to go to my working directory to make sure I'm in the right place. So now I know where the computer's thinking about. I'm going to go to this results folder and print it all out. Okay. And I'm going to call it count._files.r data. And that is a form that we can just load it back in, and our environment will repopulate. So if something went wrong or you forgot to save something, you'll just be able to pop that back open. So let's see what that looks like. Okay, we saved it, and now we're going to clean our environment with this broom over here so that our computer knows nothing. And we're going to use load function to just load that thing we just saved here, count_files.r data. And verbose equals true just means it's going to tell you that it's opening it. It doesn't need to be there really. Wow, look at that. Boom, our environment's right back to where we were. We don't have to do any of that previous work. And this will become really handy down the road. Now, you don't want to keep saving the same things over and over again, so we'll get better at that. But these are important raw data out of the pipeline that we're going to put into DESEQ. We took the salmon counts, we rounded them, we got rid of the gene name column, but archived it in our G2S, which is going to come in handy a lot. And we are now ready to make a sample sheet, and then after that's the next lesson. And then after that, we're going to start running DESEQ and finding out what genes are regulated by docs across time. All right. In the meantime, feel free to go through this video and the GitHub and the class website where it's all organized. All those links will be in the description. Okay, until next time.